Hello and welcome to this short module on inflammation. I will be providing a very basic overview of the inflammatory response and pointing out where different integrative modalities, specifically commonly used drugs and natural products, fit into this process. Let's start with a quick definition. Inflammation is a nonspecific immune response to tissue injury or the presence of a pathogen. On an acute basis, this process is essential for fighting infection and allowing tissues to heal and regenerate after some kind of challenge like an injury or even a stressor like exercise. Although we need some inflammation to stay healthy, we need it to fight infection, this process, when sustained over a long period, can damage tissues and can deregulate physiological functions. The contribution of persistent unresolved inflammation in the pathogenesis and progression of many chronic conditions has been increasingly appreciated in recent years, with applications ranging from musculoskeletal conditions to metabolic syndrome, neurocognitive decline, and even the normal aging process. So clearly there's a lot of promise in the realm of anti-inflammatories. Acute inflammation starts in the presence of an injury or pathogen. So when this occurs, there is an increase in the number of neutrophils and white blood cells that enter the tissue. And there are some neutrophils that are hanging out already in the post-capillary venule, and the bone marrow may create more of these neutrophils in response to an injury. And these cells are able to easily exit the bloodstream and enter the tissue through histamine-induced leaks in the capillary wall. And when they enter the tissue, they become active, meaning they secrete pro-inflammatory mediators and cytotoxic substances, including reactive oxygen species, which help to kill or contain the pathogen. These substances, when released over a long period of time, can create disturbances in cellular function, and they can also damage membranes and other cellular components. So let's talk about common strategies to manage inflammation. Whenever you're learning about inflammation and strategies to mitigate it, you will inevitably come across these three pathways. We have the glucocorticoid receptor, eicosanoid biosynthesis, and the NF-kappa B pathway. So we're going to walk through these three options, which collectively capture a very large percentage of currently available therapeutic agents. Corticosteroids are analogs of cortisol, which is the endogenous ligand for the glucocorticoid receptor. These drugs are powerful anti-inflammatories, but their use is limited by a number of side effects and safety issues. And those side effects and safety issues are due to a lack of specificity of this receptor for specific genes. So it has generalized effects, many of which suppress the immune system and disrupt other physiological functions. So these drugs structurally mimic cortisol, which is the endogenous ligand. It binds to the receptor, and that goes into the nucleus of the cell, and it binds to these response elements in the genome called glucocorticoid response elements, or GREs, which are located in the promoters of pro-inflammatory genes. And when the liganded GR binds, then it inhibits the production or the expression of those pro-inflammatory mediators. Another transcription factor that works very differently to induce inflammation is NF-kappa B. This transcription factor mediates pro-inflammatory gene expression, and inhibiting it can reduce inflammation. So in the absence of any kind of inflammatory trigger, NF-kappa B is sequestered in the cytosol where it's unable to function. And it's held there by a protein complex, and you don't need to know the names of the proteins. And in the presence of pro-inflammatory cytokines like TNF-alpha or chemokines, endotoxins like LPS on the surface of pathogenic bacteria, or oxidative stress, in the presence of one or more of these triggers, this sequestration complex gets phosphorylated. When that occurs, it is tagged for ubiquitin-dependent proteasomal degradation. And when it's broken down, then NF-kappa B is free to migrate into the nucleus, where it can bind to the promoters of many pro-inflammatory genes that have the response element called a K-box in their promoter. And these genes include TNF-alpha, which actually is one of the cytokines that enlists this entire process to begin with, and that could create a vicious cycle. Another gene is IL-6. Matrix metalloproteinases are also target genes of NF-kappa B. COX-2 is also upregulated. This is a very important pathway to know. Many natural products inhibit this pathway at one or more steps. Currently, there are no approved drugs whose mechanism is direct NF-kappa B inhibition, but natural products often act as low affinity inhibitors of one or more steps that we won't go into here, but just recognize that NF-kappa B inhibitors are very diverse in structure, and they mainly include the polyphenols and the terpenoids. So polyphenols include curcumin, resveratrol, quercetin, and catechins from green tea. And the terpenoids include boswellic acids from Boswellia serrata, which is also known as Indian frankincense, and other triterpenoids present in rosemary and other culinary and medicinal plants. The NRF2 pathway, which we won't cover here, is also inhibitory towards NF-kappa B. So if you activate NRF2 with something like sulforaphane, then you can indirectly inhibit NF-kappa B. NRF2 or NRF2 activators are gaining a lot of traction in the research literature as promising strategies aimed at reducing inflammation. If you've ever studied pharmacology before, you have probably seen these pathways of eicosanoid biosynthesis, and these are important pathways to understand, not in great detail, but just be familiar with them. Non-steroidal anti-inflammatories intersect with this pathway, and a variety of other agents like fish oils and certain phytochemicals modify these reactions. So on the left, we have a pro-inflammatory cascade that's initiated when phospholipase A2 or PLA2 converts membrane lipids into arachidonic acid, and that is the precursor for the inflammatory leukotrienes, series 2 prostaglandins, and thromboxanes. Non-steroidal anti-inflammatories inhibit COX, which is responsible for the conversion of arachidonic acid into series 2 prostaglandins, which are directly responsible for most of the cardinal symptoms of inflammation: redness, heat, swelling, and pain. Leukotrienes are therapeutically interesting because in conditions like asthma, you end up with a lot of leukotriene production. On the right-hand side, we have an anti-inflammatory cascade that is made possible through omega-3 fatty acids, which must be obtained through the diet. Linolenic acid is converted to EPA, and EPA is then converted to DPA and DHA. And these three compounds are precursors to specialized pro-resolving mediators, or SPMs, which include the resolvins, protectins, and muresins. These compounds trigger and execute the resolution phase of inflammation, which we will expand on in a moment. Just a quick note on a phytochemical called acetyl-keto-beta-boswellic acid, or AKBA. This is from Boswellia serrata, also called Indian frankincense. AKBA is an allosteric inhibitor of 5-lipoxygenase and thereby reduces the formation of leukotrienes. So now that we've covered the basics, let's just review each category. We have corticosteroids that work through the glucocorticoid receptor. We have eicosanoid modulators, which include non-steroidal anti-inflammatories, triterpenoids like boswellic acids, and essential fatty acids like EPA and DHA. And then we have curcumin, resveratrol, and other polyphenols and phytochemicals, which are all NF-kappa B inhibitors. So now that we've reviewed this, let's go back to the inflammatory response and finish the story. The resolution of inflammation begins when the neutrophils reach the end of their life and they begin to release these substances called SPMs, or specialized pro-resolving mediators. And SPMs are the protectins, resolvins, and muresins that I mentioned as metabolites of the omega-3 fatty acids. These SPMs inhibit further infiltration of neutrophils. They also dampen the effects of pro-inflammatory cytokines and other signaling molecules. Some of them actually inhibit the NF-kappa B pathway. When macrophages ingest the SPM-emitting neutrophils, they transform into resolution phase macrophages. So this designates their phenotype to be strictly cleanup duty. And not only that, but they can set the stage for healing and regeneration. So those macrophages are very specialized. They're able to clean up dead cells, lingering bacteria, and they actively promote a return to homeostasis. So historically in pharmacology, we have focused on agents that inhibit the initiation process. Now we know a lot more about the bigger picture. So new therapies are being developed to address and support the resolution phase, knowing that chronic inflammation is likely to be a failure of resolution and not so much strictly a perpetuation of the initiation process. I hope this has been helpful in bringing you up to speed on a very complicated topic. If you'd like to learn more, I've included a few links to open access articles. I've also included a few study questions and an answer key if you'd like to test your knowledge. Don't forget to check out the other learning resources at integrativepharmacology.com, and don't hesitate to reach out to me with questions, comments, or suggestions.